Okay, so now, yeah, to some fun stuff. So single cell alignment kind of works the same way. So we get, in the end, we get a FASTQ file, and we just have to find where it came from on the reference with like slight modifications to it. And the slight modifications are is that every read has a barcode and a something called the UMI, which is a unique identifier for that sequence. And so this all started with this technology called like drop-seq. Okay, so the idea is the following. So you have your you have barcoded beads. So we know these already from L1000, right? And so they're covered in these barcodes. And we try to send them one by one through this pipe. And then we intersect them with a stream of cells that are suspended individually. So hopefully they're like just by themselves. And then most of the times they don't meet. It's, like an intersection where everybody drives through the red traffic light and usually nothing happens. But then sometimes two of them will meet. And, they go through here and then they get encapsulated in this droplet, it's called. So then you have a bead and a cell in one droplet. And so with the modern technologies, they can do all these processes, like all these multiplication and sequencing and whatnot in this droplet. And so what happens then is that all your reads that come from this droplet or like from the cell have this barcode on it, right? So then so then everything when you find a sequence that has a certain barcode, it comes from that cell, right? You can kind of group them together that way. So, but then, yeah, you mostly get empty droplets, and then sometimes, a cell and a barcode, they meet together. Sometimes you have two cells. So that's what you try to prevent. So that's why most of them are empty. Okay. So this is kind of how this looks like. So if imagine this is our FASTQ file, and then we have the cell barcode. So this is, just a sequence, and then this is synonymous now for a cell. So if we see this sequence again, then we know that these reads, whatever comes afterwards, is from the same cell, right? So like the first step of the algorithm would be to find the barcode and then find the location of the sequence on the reference, right? And then allocate the count to the gene for that cell. But yeah, let me see. Right, so this is exactly how this works. And so in 10x genomics, what they do is they do paired end reads. On the one read, you have the barcode and the UMI. I'll explain the UMI in a bit, but so on the one read, you have the barcode and the UMI, and in the other one, you have the gene. And they actually recommend that you only sequence, I think it's 26. I think it's 26 base pairs. I think the UMI is 10 base pairs and the barcode is 16 base pairs. But oftentimes people don't read the protocol, I think, and like they just sequence both to like 100, so they waste a lot of money sequencing the left reads too deep, but too long, but something to consider. Right. Right, so this is really the workflow, how it looks like. So what it does is it takes all the reads, right, and then it looks for barcodes, right? So you have a list of barcodes in a text file. So I think there's like 50,000 or something. So you could have technically 50,000 single cells because that's the amount of barcodes you have. And then try to find where the read maps. If it doesn't map, you discard the read. Or if the quality of the reads is too low, you discard the read. And now with whatever you have left has a barcode and it aligned to a reference, right? And now, interestingly, so in Cell Ranger, which is the one that is from like 10x genomics, they align also against intergenic reads. So they actually also count introns as a valid alignment target, which is interesting because it's not mature mRNA. But so there you go. And they they also dismiss anything that isn't uniquely mapped, if I understand it correctly. And they always map against a gene. It's not on a transcript level, it's only on a gene level, because you don't have enough reads to be confident about transcript level stuff, right? So now we have a read that aligned somewhere on a gene sequence, is uniquely mapped, has good quality of reads, and has a barcode. And so now it also looks at this UMI. So the UMI sequence is 10 base pairs after the barcode. And the UMI is it's just like a random unique sequence that tells you, so you use it because you're worried about when you replicate the library that you have, that you skew towards like one read that just gets like multiplied over and over again. So you overcount that read, even though it's the same, if it's from the same mRNA. So you don't want to count them double, right? So this is what that UMI is for. So in the end, you only count unique UMIs per gene. You don't count the actual occurrences of a read. And yeah, so that gives you the UMIs. And then from there, you have to decide where you cut off if a single cell exists. So if a barcode is valid, there should be a certain amount of unique UMIs attributed to that barcode. And then, so in this case, they decide that there's about 1,100 cells that they detected, and then it outputs a matrix of gene counts. And so running out of time. Okay, so the web summary of this is pretty nice. So it tells you what the expected number of reads of cells is. So it says it found 6,233 cells. It found 12,000 reads per cell on average, which isn't a lot, right? It's kind of surprising how little that is. And then it finds, when it says median genes per cell, it basically means like as soon as a gene has a single read, it's detected, right? So that's what that means. So that's kind of what we get. And then here you see the UMI count. So there was one cell that got 100,000 or more than 100,000 unique uh, mRNA fragments. But the one with the cutoff is more at like 1,000, right? So there's a widespread of coverage for these single cells, right? And so you can see this pretty quickly in this summary. And it will tell you, how many reads aligned. So I gave it like 75 million, and then most of them had a barcode, stuff like this. You'll find that all here. And then an interesting one is, so then it does like some clustering, which will be the next lecture, where you can see how the single cells cluster. You'll find some interesting genes that, are unique to the clusters. And then, so here, these are kind of interesting. So this is the sequencing saturation and median genes per cell. And so these plots will give you an idea if you were too stingy when you decided how you sequence your data, I guess. So this plot here will tell you the fraction of duplicated UMIs. And so basically what this tells you is if you would increase the amount of sequencing you do, would you get more information out of it, right? And so this dotted line is where it anneals to, I guess. So you could never get better than, in this case, like 0.9. And so this what this fraction of UMIs means, if you duplicate them, let's say in this case, at a mean reads per cell of 5,000, 20% of the UMIs were duplicated, right? So meaning most of them were unique. If I would do more and more, eventually I would just find stuff I already found, right? I just sample from a pool of things, and eventually I've seen it multiple times. I'm like, it's like collecting cards, and like you have nine and you need the last one, but like you'll never draw it, right? So it's not worth it to go on. But so this is kind of showing you this, that you could have, you could have done more sequencing and you would have probably gotten more information out. That's just what that plot means. This is somewhat similar. Okay, and then so the outputs will be these three files. The important one, well, they're all important, but so the matrix MTX is this text file, and it has these three columns. The last one is the actual count. The second one is I should have written this down. I think the first one is the features. So this would be relative to, this is like the number of features. So there's like 36,000 genes. So this is the gene 1725 relative to the features row. So the features is just the genes, and then this refers to the row in that feature file. And then this one is the cell, I think. I don't know why it says 700,000. I'm pretty sure this is the cell. So this is kind of, oh, never mind. This is the barcode. So but this is the same as to say it's cell 4, cell 5, cell 6, right? But not all the barcodes are there. And so I'm pretty sure this number used to be the number of unique barcodes that it found. But this is the maximum number of rows in the barcode. So I checked this. I don't know if this is a bug in the latest version, but this just tells you the length or like the number of barcodes in total, not the number of barcodes it actually detected. And yeah, so this is how you run Cell Ranger. And so I left out how to build the index because you can download it from their website. So it's easier than building it, or there might be steps involved that you can't even easily replicate. So you can download the index and then you just run the alignment. So one thing that's unusual, yeah, you need a lot of memory. They say at least 8 CPU cores. And then when I tested it, like on my system, like if you want to do 100 million reads, it takes about an hour. That's like the ballpark. And then you need, because it's paired end reads, as I said, you need this kind of convention of naming your reads. Otherwise, it doesn't work for me. But there's like extensive documentation on their website. But to keep it simple, this is kind of what this looks like.